Welcome to the second session of this year's AGM for the Coronavirus Network. It's great to, and this session, as you'll have known from your careful studying of the programme, is on Betacoronaviruses. I am under very strict orders from Professor Diggard, who we treat with respect here, to make sure that all of the speakers keep to time. So I know the first one's going to do a great job there. The first speaker is Grace Tyson, who is going to introduce herself and her topic and take the microphone from me.
Sure. Okay, so I'm Grace. I'm a PhD student at the University of Glasgow, and today I'm going to talk a little bit about immunity to SARS-CoV-2 in companion animals, primarily cats. So we know that whenever there are cross-species jumps of viruses, it can be dangerous. It can introduce new animals as reservoirs, and it could potentially lead to the emergence of new variants. And we also know that SARS-CoV-2 is present in a lot of different animals. It's able to infect a lot of different animals. Most of these species are infected from an anthropogenic origin, humans passing to their pets or to other animals, but in some species there is evidence of interspecies transmission. So we've seen that in mink farms and in white-tailed deer in America. But this, in my opinion, is particularly concerning in companion animals such as cats and dogs because they live in such close proximity to humans. So you can get transfer potentially back and forth. So for our study, we wanted to do a serosurvey to determine what the prevalence of SARS-CoV-2 is in UK cats and then also in UK dogs. And we also wanted to further characterise this feline and canine immune response against different variants. So we did this using a pseudotype neutralisation assay. We generated pseudotypes bearing the spikes of all the different SARS-CoV-2 variants that we wanted to test. We tested on a fixed dilution assay, and then we titrated the positives to ascertain the actual strength of the neutralising antibody response. And then we did an ELISA. So we did the ELISA because not only it detects non-neutralising antibody, but also it will confirm whether we were correct in our neutralisation assays. So we were very kindly provided a serum from the University of Glasgow's Veterinary Diagnostic Service. This was all residual serum that was sent for other tests, and we got the leftovers of it. And all of the animals were from the UK. We started with cats. We got nearly 6,000 feline samples. These were generally pet cats or cats from shelters. And then we started adding pet dogs as well. We have 812 of those. Our samples are from all the way throughout the pandemic up until January of 2024. So I'm not going to go into this because Hagar's already spoken about this. But for our neutralisation assay, we used a pseudotype panel of 8 different viruses, each bearing the spike proteins of a variant of concern that was dominant at some point in the UK. So this was the ancestral strain, Alpha, Delta, Omicron, and then BA2, BA5, BQ1.1, and XBB, which are all derivatives of Omicron. And we could characterise our samples as positive neutralising, positive non-neutralising, or negative if they're negative on both the ELISA and the neutralisation assay. So as you can see, there's been a rise in feline seroprevalence over time. It's risen throughout sort of 2021, and then it has kind of plateaued. So we have a 4.8% neutralising seroprevalence, and when we include the ELISA-only samples, it's nearly 14% in UK cats. And we can break this down by dominant variant and compare it to human data. Now this graph is a bit confused, so if we break it down by variant, what you can see is that the emergence of variants into cats is after the emergence of the variant into the human population. So we can see that most of these infections in cats, they appear to be coming from humans into cats, from owners to their pets. So the solid line is the sequences in humans, and the line graph is dominant variant in cats. So dominant variant, what we did was we organised our samples by which variant they had the highest titer against, and this is the result. What we can also see is that immunity remains long after the variant has become extinct, and this is what we expect with antibody immunity. But it is waning over time in some of the variants. And the other thing we can see from these graphs is that as Omicron and its descendant variants appear, they appear to be less immunogenic. The peaks in feline immunity are no longer as high, they're no longer as defined. And when we look at cats versus dogs, we can see that they actually have quite a similar seroprevalence, 4.8% neutralising for cats and 4.4% neutralising for dogs. So cats had a slightly higher seropositivity, but not significantly. But what we can see is that cats had significantly higher both mean and highest or dominant titers than dogs. We don't know why this is. It could potentially be due to our sample collection. As you can see here, our cats were more likely to be infected with, or likely infected with, a pre-Omicron variant, which leads to a stronger immune response. So some of the risk factors that we were able to look at, we wanted to break them down by things like breed and age and things like that. So we realised that pedigree cats have a higher seroprevalence than non-pedigree cats, but breed has no effect on titer. We also found out that sex has no effect on either seroprevalence or titer. Both male and female cats had around the same seroprevalence. Again, neuter status had no effect on seroprevalence or titer, but we did find that cats aged 2 to 5 in this kind of young adult range had a higher seroprevalence, but no difference in titer. We had another look at the breeds of cats in our study, and we looked at the major breeds, and of all the pedigree breeds, Burmese and Bengal cats have the highest seroprevalence. Now, it should be noted, pedigree cats, they're estimated to represent around about 10% of the UK's feline population. Most cats in the UK are moggies, but the feline population of this study was around about 33% pedigree, so it was slightly skewed, and the data set also skewed young and male. So an interesting thing that we found from this is when we categorise our samples by their dominant variant, the variant which they had the highest titer against, we can see that in pre-Omicron variants, there was quite good cross-neutralisation of all of the pre-Omicron variants. So if you had your highest titer against alpha, then it was likely that the sample was also able to neutralise the ancestral variant and delta. But as we moved through into Omicron and its derivative variants, it kind of changed. And there was a definite delineation between pre-Omicron immunity and post-Omicron immunity. So if your cat had a higher titer against a post-Omicron variant, they were more likely to have a very, very low titer or no titer against pre-Omicron variants. And we can look at this further. So all of these samples had their dominant titer as a pre-Omicron variant, and you can see how low the post-Omicron immunity is there in both cats and dogs. And all of these samples had their dominant variant as a post-Omicron sample, and you can see how low the pre-Omicron variant is. We also looked at it statistically, and you can see where the red is, that's the highest correlation. So we're definitely seeing this trend there as well. So yeah, like I said, probable infection with the pre-Omicron variant provides this broad neutralising immunity against pre-Omicron variants, but with post-Omicron variants, you're likely to only get immunity against post-Omicron variants. And this is comparable to some findings in naive humans. We've seen that Omicron immunity, it kind of manifests differently. So why is this? So we know that Omicron didn't actually evolve chronologically as the previous variants did. It didn't go ancestral alpha, delta, Omicron. Its closest ancestor was in mid-2020. We don't 100% know why this is or how it emerged, but there's 3 theories. One is that it circulated in a population with low surveillance, and we just didn't see it. One is that it evolved in a chronically infected patient, which had a sort of environment conducive to mutation. And the other theory, it's been posited that Omicron emerged due to a species jump, but we don't know why. However, we do know that these mutations caused quite a distinct change in spike and RBD in Omicron from the ancestral strain. And then the emergence of Omicron subsequently led to the emergence of lineages like BA2, BA5, and others. So does this genetic turning point translate to serology? And is it time for serotype 2? Well, I'll let you decide, but we know that there are distinct pre- and post-Omicron immunity profiles in companion animals. There's also a lot of evasion of ancestral strain derived immunity and ancestral strain vaccine derived immunity. We can see that there's imprint-dependent boosting of pre-Omicron vaccines. There's low neutralization of pre-Omicron variants in Omicron-infected individuals. And when you put titer results into antigen mapping software, generally Omicron and its derivatives are classified as a separate serotype. Now, serotypes, they're difficult to characterize in humans because we've all been infected so many times and vaccinated and all sorts. But in my opinion, I think Omicron does have some signs of a separate serotype. So some conclusions. We have a feline seroprevalence of 4.8% neutralizing and a canine seroprevalence of 4.4. There's no significant difference between the two species. Through serology, we can see these mirror waves of human infection in cats. It looks like the viruses have jumped from humans into the feline population. Omicron and its descendants appear to be less immunogenic in both cats and dogs than pre-Omicron variants. Pedigree cats and cats in the 2 to 5 year age group are risk factors for seropositivity of SARS-CoV-2 and have higher rates of seropositivity. Lower SARS-CoV-2 seroprevalence in dogs and cats, however, this isn't significant, but there are significantly lower titers in dogs than in cats. And there's lower levels of cross-neutralization of Omicron and its descendants by pre-Omicron derived antibodies. So I hope from this you can see that SARS-CoV-2 remains quite an underappreciated problem in companion animals, and adoption of a One Health strategy in coronavirus surveillance is pretty essential when we're looking at these species jumps. So I just want to say thank you to everybody who helped with this, and to all of you for listening. Thank you very much.
Thank you very much to Dr. Tyson, and we've got 3 questions. Dr. Diggle first.
Great talk. Do cats get sequential infections then, like humans?
So we don't know. We haven't been able to actually PCR test cats sort of longitudinally. I'd like to have done that. I don't see why not. We have a few samples that we had multiple samples from the same cats due to labelling errors, and it does seem that immunity tends to wane over time. So it's absolutely possible. And especially when we look at the pre-Omicron and post-Omicron immune profiles, I absolutely believe they could be reinfected with a later lineage of virus.
So why 2 to 5 are more susceptible than, say, 1-year-olds? It's anything to do with maternal antibody? Why is
I'm not sure. My inclination would be to say that, you know, cats aged 2 to 5 are kind of in their prime. They're more likely to be interacting with other cats. So it may be cat-to-cat transmission, but I'm really not sure as to why exactly that is. But it's something I'd definitely like to look into.
Cool, thank you. There's a question up on the right there.
The other one might be.
You're not allowed to throw that.
Great talk. I was just wondering, you describe Omicron as being less immunogenic in cats and dogs than the previous variants. Is there any evidence regarding Omicron's ability to infect cats? Like, could your data be explained by there are actually just fewer infections of Omicron in cats than the previous variants?
So it's possible, but you know, we're collecting variants across the pandemic, and we see titers that are lower in apparently Omicron-infected cats than, you know, we're getting, for example, samples collected in 2023 where the cat has obviously been infected with something like alpha and the titers are still really strong, but ones that are collected during the Omicron wave and they only have an Omicron titer and it's really, really low. So I do think there is a difference in immunity to it. There may be fewer infections, I don't know, but I do think there is a difference in immunity.
Thank you. So you did mention at the beginning that the transmission in deer and mink has also been seen, and we have seen transmission back into humans, but have we seen them with feline dogs? And also evidence of accelerated evolution in the deer back to humans has been established, but perhaps, I don't know if you've seen that in your data set.
So there's been a few different cases that we know of infection in felids that have been transferred back to humans. One was a lion at a zoo in Indiana, and one was a cat in Thailand that transferred the virus to their vet. It's not really closely, there's not close surveillance of it, so we don't know. Probably there are more cases, but we have seen that. I can't remember the second part of your question, I'm sorry. I'm sorry, I can't remember, but yeah, there have been cases in cats, yes.
Thank you very much indeed, Grace, for a really interesting talk and great questions. I'm now going to hand over to my co-chair, and we're going to alternate.